The cardiovascular system is made up of three components: blood, the heart, and vessels. In this chapter and video, we're going to focus on blood. This is the medium of transport, but that's not its only function. We study and analyze blood because it helps us detect many different types of infections and diseases. And it also helps us determine if an individual is healthy or not. Other functions for the blood include protection, so it has a role in immunity through the action of many different white blood cells. It also has a role in clotting, which protects against excessive loss of blood from the cardiovascular system in case of injury. We know that its major function is in transport of gases and nutrients, as well as other solutes and hormones, to cells that are stationary and can't get these things on their own. And another major function is regulation. One thing that the blood regulates is pH by the use of buffers, so it maintains the pH. It also regulates body temperature because of the properties of water, which we know is present in blood, and also maintains the water content of cells because of its specific concentration of solutes and proteins that maintain blood osmolarity. In this first section, we're going to focus on red blood cells and the major properties of blood. Blood in general is a liquid connective tissue. It's made up of cells that are surrounded by a liquid matrix. It's different than the interstitial fluid that surrounds the cells. However, there's interaction between the two. Blood always supplies the nutrients and gases to the cells through the interstitial fluid. So these components will move from the blood into the interstitial fluid and then into the cell. And the opposite is also true for waste or carbon dioxide. The cells are going to transport these things out of the cell into the interstitial fluid and eventually into the blood. If we centrifuge a vial of blood, we see that it can separate into different layers. The bottom layer, we get what we call formed elements. And on the top layer, we get what we call plasma. Plasma makes up 55% of blood, and it's mainly made up of water, proteins, and other solutes. The rest, the 45% that's left, is those cellular components like red blood cells, white blood cells, and platelets. You can use this table here as a summary of all of the components of plasma, where they're made and their function, as well as for these functions of the formed elements. We will cover all of these separately, but this is a good table to always go back to. A few more characteristics of blood is that it's thicker, denser than water, and it's also slightly sticky. Its pH is between 7.35 and 7.45. Its temperature is 1 degree higher compared to body temperature. It's around 38 degrees Celsius or 100.4 degrees Fahrenheit. And depending on the oxygen concentration, the color is slightly different. If it's completely full of oxygen, it's saturated, there's a lot of oxygen in the blood, it looks bright red. However, if there is more CO2 and less oxygen, it looks dark red. So let's focus on plasma next. We know that this is the watery component of the blood, but it does have dissolved solutes and nutrients, as well as hormones, enzymes, gases, and the rest. Blood is around 55% plasma. And when we focus on plasma itself, it's mostly water, and specifically around 91% of plasma is water. The rest are proteins and solutes, but we definitely see more proteins here. And these proteins, some of them are confined to the blood, so we call them plasma proteins. Some of them are made in the liver. We're talking about albumin, fibrinogen, and globulins. These are the three major plasma proteins. And then we have those solutes that include electrolytes. We have enzymes, hormones, gases, and waste products like urea, uric acid, creatinine, and bilirubin. Let's talk about those plasma proteins and their function in the blood. Around 7 to 8% of the plasma is plasma proteins. Those are the three major ones. Like I said, they are synthesized or produced in the liver. Albumins would be the smallest, however, the most abundant plasma protein, and its major function is to help maintain osmotic pressure. This is going to be a protein that assists in the exchange of fluids, especially in the blood capillaries, and it creates this environment that makes the osmotic pressure high, so it can draw water from the tissues into the capillaries to maintain blood volume and pressure. And again, this is the most abundant plasma protein. Globulins are large plasma proteins and have different functions depending on the type. Alpha and beta globulins are involved in transport. Gamma globulins are what you know as antibodies. And so we know antibodies have a function in the immune response, attacking viruses and bacteria. So these are specifically secreted or made by B cells and participate in protection. The last plasma protein that I want to mention is fibrinogen. It's also a large protein, and this one plays an important role in blood clotting and preventing additional or excessive blood loss. And then we have the formed elements of the blood, and those include three major components: erythrocytes, which are also known as red blood cells, we can call them RBCs, leukocytes, known as white blood cells, and the last components are platelets, also known as thrombocytes. We call them cellular components because not all of them are essentially cells. For example, platelets are fragments of cells and not complete cells. Red blood cells and white blood cells are, on the other hand. The next topic in this video is what is a hematocrit? A hematocrit measures the percentage of the amount of red blood cells that is present in whole blood. And this is usually indicative of certain conditions in the body, especially if the values deviate from the normal range. The normal hematocrit for an adult female is between 37 to 47%. And so we're talking about that number being the percentage of red blood cells in whole blood. So once you centrifuge it, spin it down, 37 to 47% of it should be those red blood cells, and the rest should be plasma mainly. For males, it's around 42 to 52%. So any value that is lower than 37 or higher than 52 indicates anemia or polycythemia respectively. Anemia refers to a depressed hematocrit or low percentage of red blood cells. This is lower than normal. And polycythemia refers to elevated red blood cell percentage. We discussed formed elements or these cellular components, but what is their origin? Where are they produced? The site of production for blood cells, all of them, is the red bone marrow. The process that leads to their production is called hemopoiesis or hematopoiesis. The red bone marrow is the primary site for this process, and it's the primary source of blood cells after birth and throughout life. The red bone marrow has a lot of vessels, and it has microscopic spaces between the trabeculi of spongy bone, and this is where we find these pluripotent stem cells that will divide and differentiate into the different types of blood cells that we have. This whole process is stimulated, of course, by hormones, and the number of red blood cells and platelets should remain steady. However, the number of white blood cells will change depending on the state of the body, whether it's undergoing infection, cancer, or it's being exposed to other foreign antigens. Let's take a look at the process of hematopoiesis. In order to form blood cells, those pluripotent stem cells in the red bone marrow produce two further types of stem cells. And these then have the capacity to develop into several types of cells. The two main types of cells that originate here are the myeloid stem cells and the lymphoid stem cells. And these are going to differentiate into the different types of blood cells that we have. Let's start with the myeloid stem cell line. This one leads to the production eventually of red blood cells, platelets, basophils, neutrophils, eosinophils, and monocytes, which eventually become macrophages. On the other hand, the lymphoid stem cell line leads to the production of lymphocytes like T and B lymphocytes, as well as a natural killer cell. Like I mentioned, hormones stimulate the process of hematopoiesis. These hormones are collectively called hematopoietic growth factors, and depending on the hormone, we're going to see an increase in a specific type of cell. If we need more red blood cells, erythropoietin is secreted, or EPO. If we need more platelets, then thrombopoietin is secreted, or TPO, thrombo for thrombocyte. And cytokines are secreted to stimulate the increase of the number of white blood cell precursors, which eventually lead to more white blood cells. Like we mentioned, with formation of blood cells, we have two main stem cell lines, the myeloid and the lymphoid. And depending on which line is stimulated through which hormone, we get the specific types of cells that we need. For red blood cells, the process of their production is specifically called erythropoiesis, which begins in the bone marrow. Eventually, the myeloid stem cell line gives rise to blast types of cells, which will commit to forming these red blood cells. Through that process, a cell called a reticulocyte is produced. This is an immature type of red blood cell that is just kicking its nucleus out. So it's just ejected its nucleus. It goes into the circulation, and in 1 to 2 days, it becomes a mature red blood cell. For red blood cells, they need to get rid of their organelles and nucleus so they can fit as much protein as possible and oxygen as well. Erythropoietin, the hormone that stimulates its production, is formed in the kidneys. And this is usually due to low amounts of oxygen in the blood, known as hypoxia. So this is detected, and then EPO is secreted. Anemia or low blood cell count can initiate EPO secretion as well. So some major characteristics for erythrocytes: they are flat and biconcave disc-shaped cells that carry oxygen. They don't have the mitochondria or a nucleus, and for those reasons, they can only survive around 120 days. They pack lots of hemoglobin, around 280, and their function is to transport oxygen, carbon dioxide, from and to cells. The hemoglobin portion of the red blood cell is the protein that is found there, and that's where the oxygen is carried. When oxygen is bound to hemoglobin, we have what we call oxyhemoglobin, and this usually has 4 binding sites for oxygen. So let's take a look at a molecule of hemoglobin here. In the image, take a look at how many protein or polypeptide structures are found in the hemoglobin protein. It has 4 different chains, the alpha chain and the beta chain. They are arranged in a specific conformation, which allow the hemoglobin to have its structure and then function. And in the middle of each chain, we have what we call a heme group. This heme group has an iron right in the middle, and this is where the oxygen is going to bind. This structure can bind 1 oxygen, and since we have 4 of them in a hemoglobin molecule, then each hemoglobin can pack 4 oxygens. And then think about each cell having around 280 million, right, hemoglobin molecules, and each of those is carrying 4. That's a lot of oxygen bound to hemoglobin in each red blood cell. This is what it looks like when it's bound to oxygen. We call it oxyhemoglobin. It's also important to note that hemoglobin can bind to carbon dioxide. And so we get carboxyhemoglobin and carbaminohemoglobin. Hemoglobin has other functions as well. It has nitric oxide, which is a gaseous hormone bound to hemoglobin. And once hemoglobin releases nitric oxide in the circulation, then nitric oxide will cause a vasodilation, or the increase in the diameter or expansion of the blood vessels to improve blood flow to a specific area. So it can regulate blood flow and blood pressure by secreting this gaseous hormone we call nitric oxide. Nitric oxide is involved in vasodilation. That's what it stimulates, and you can see here, once the red blood cell secreted nitric oxide, then we see a little bit of an expansion there to allow the blood to move through. Under specific conditions, when we have mutations in the gene that leads to hemoglobin production, we get sickle cell disease. Sickle cell disease refers to an abnormal shape of a hemoglobin, which causes the cell to take up a sickle or crescent shape, which doesn't allow it to have its flat biconcave shape. We know that that shape allows it to squeeze through in the vessels and carry oxygen. In this specific state, the oxygen carrying capacity of blood goes down, and it's not as efficient delivering oxygen to cells. It also depends on how severe the disease is, and if a person has only 1 copy of the mutated gene or both copies. In order for a person to have sickle cell anemia, then both copies need to have the mutated gene or allele to exhibit this disorder. Red blood cells, like I mentioned, only survive around 120 days, and when it's close to that time, They are removed from the circulation by macrophages in the spleen and the liver. And these macrophages have phagocytic capabilities, which means that they can break down the red blood cells. Some of the products we can get rid of. Some of the products are recycled and reused. Which part is recycled? The iron heme structure is recycled from the liver and the spleen. The iron is bound to transferrin, which is going to be a structure that binds to iron so it can preserve it in the body, and it takes it to the red bone marrow where new red blood cells are produced. Part of the heme group is broken down though and transformed into biliverdin, which we transport to the liver. And then in the liver, it's converted to bilirubin, which we know is eventually used to make bile, and then transferred into the small intestine. It's not used. Eventually, it's excreted as waste. Any amino acid components from the hemoglobin protein can be reused in the production of new red blood cells or erythrocytes. One last component that is found in the red blood cell is carbonic anhydrase, which is an enzyme that converts carbon dioxide and water to carbonic acid. Carbonic acid and bicarbonates are the compounds that transport around 70% of the carbon dioxide. It allows it to dissolve in the plasma in that way and transports it to different locations in the body. Bicarbonate also serves as a buffer. And so the catalyzed reaction looks like this. So CO2 and water are combined through this enzyme to give us carbonic acid, and carbonic acid will dissociate into H+ and bicarbonate in the plasma. Recall that bicarbonate also serves as a buffer in the blood. It can bind to the H+ when there's too much H+ in the bloodstream. But most of the CO2 is transported in the form of bicarbonate, which allows it to dissolve in the plasma and circulate that way. In this video, we discussed the different properties of blood production, and characteristics of red blood cells.